Las personas que están en el fondo sur, que se acerquen un poquito. Okay, hello everybody. So today we have with us Fernando Alda. Many of you know him because he did his PhD here at the museum. He did his bachelor in the University of Navarra and came here to Madrid for his PhD in 2009, where he worked with Doadrio, but he didn't work on fish by that time. Then he went for postdoctoral work to the Smithsonian Tropical Research Institute in Panama, where he did work on fish and other stuff. And then he did another postdoc in Tulane University and another one in Louisiana State University. And then he got in 2019 an assistant professorship at University of Tennessee. And then in 2023, he finished his US adventure and got a position as científico titular at IREC in Ciudad Real. And he works on systematics, population genomics, and biodiversity. He integrates field and molecular work. He also integrates museum collections. He integrates as well ecological and geological perspectives to investigate genetic variation and speciation across multiple biological scales. So he works on many of the topics that we do work here: genomics, museomics, systematics. He's a good candidate to work in our department. He has worked in different number of taxa, rabbits, lynx, raccoons, viruses, but also fish. I think today he's mostly going to be working on fish. He mostly works nowadays in ultra-conserved elements, but also mitochondrial genomes. And he has some very cool papers in systematic biology, for example, and a very recent review in Molecule that I hope to see. And I leave you with.
Thank you. Thank you very much, Marta. And thank you, everybody, for being here. It's very nice to see so many friendly faces and be back here at the museum. That makes me very happy, but also a little bit nervous, but it's all good. All right, so today I'm going to be talking a little bit about my journey on this world of systematics and applying phylogenomic methods to try to resolve fish radiations. And most of these, I'm using fish as a model, but could be applied to many of the organisms that we all work with. And I don't know if you're familiar with Dr. Seuss. He's a very famous author of children's book in the US. Here, maybe he's more famous for being the author of The Grinch or other books. But it's actually kind of cliche when you're talking about fish to refer to his book, One Fish, Two Fish, Red Fish, Blue Fish. So I apologize for being so unoriginal. But reading through this book, I found that some of the things that Dr. Seuss wrote actually match pretty closely what I work with, or what we all work with, and I took the liberty of only editing a little bit his work. And one of the pages of the book would say something like this: is that say what a lot of fish there are. Some are red and some are blue. Some are old and some are new. Some live deep. Some are sheen. Some are very, very mean. Why are they deep and sheen and mean? I don't know. Go ask their genes. So in the end, this is kind of what we're trying to do here, right? We're trying to discover or understand how many of these behaviors or phenotypes or distributions have evolved, and using molecular phylogenies or molecular data to understand these patterns and these processes and mechanisms. So basically, this is the outline of my talk. I'm going to be talking about kind of like a mean group of fish, including the electric eel. I'm going to be also talking some examples about some pretty colorful fish like this Trichromis here, and also some other fish that live in very extreme environments in the depths, like this Lamprologus cichlid from the Congo River. So, as Marta was saying, I wasn't working with fish when I was here doing my PhD here at the museum, but of course, working with in Nacho's lab, or there's something that was that stayed there. And most of my interest in fish started basically related to how useful they are or how good models they are to understand phylogeographic or biogeographical patterns. And one of these groups of fish that are interesting to understand this broad-scale biogeography could be, for example, this Osarophysin fish, which is the second largest superorder of fish, the largest one. 70% of the freshwater fish belong to this group. They have these widespread distributions across the globe, and some of them pose interesting questions because they allow us to test, for example, the roles of vicariance and dispersal in driving these the evolution of these groups. For example, in the case of the Carassiformes, these two suborders, the Caracoidae in the Neotropics and the Ciceroniidae in Africa, right? So interestingly, in this group, despite how well known most of these fish are, there are still a lot of questions that we don't know yet who's more related to who. We don't know if a tetra is more related to a catfish or to an electric knifefish. And this was one of the first projects that I got involved when starting to work in phylogenomics. And at this time, I just started my postdoc at Louisiana State University at LSU, and there I was working with Prasanta Chakrabarty and Brant Fairclough, who's one of the main developers of ultra-conserved elements, and they were proposing to use phylogenomics to resolve this issue, right? It's like, oh, we're just gonna add a bunch of sequences, a bunch of genes, and we're gonna solve this question. And back in the days, this was me like, oh, this is gonna be easy. I've been just like sequencing mitochondrial genes, and phylogenomics is going to resolve the issue, but that was not the case as you would expect. And one of the unexpected results that we got in this first project is that we did not recover the Carassiformes as a monophyletic group. This was kind of striking, not only because of the biogeographical implications, but to invoke the non-monophyly of Carassiformes, we would also need to try to reconcile some extreme convergence of characters, like teeth characters between these two suborders, and so forth. So it's like, and I was surprised, like, well, what is going on here? How are we going to explain this? Looking more closely and me starting to learn of what was going on here, we started looking at some problematic areas of these trees, particularly these short branches that could result in this terrible and scary thing that is called the anomaly zone, right? So what is the anomaly zone? The anomaly zone is anomalous gene trees actually are when using methods of maximum likelihood in concatenation, we could recover that the most common gene tree is actually not the species tree that we're recovering. So not the most frequently recovered gene tree is not what our concatenated phylogeny, that is all our genes analyzed all together, are recovering. And this is, in a nutshell, a problem of short branch lengths. The shortest this preceding branch length is, it increases exponentially the probability that we are going to find one or more anomalous gene trees. So gene trees that do not match the final species tree that we're recovering. Anyway, so this is more important than just non-recovering monophyletic Cruciformes. I guess very few people here care whether these fish form a monophyletic group or they don't. But right when we were working on this, other groups and other papers came out, coincidentally working with the same group of fish that were also asking these type of questions. How can we interrogate genomic data to resolve these conflicting groups or these recalcitrant relationships in the tree of life? What these studies showed is that basically when we have a large genomic data set, it is possible to basically or virtually recover every possible relationship with one of the markers that we're analyzing. And this goes back to this idea that I really like, is that seeing phylogenies or species tree as a cloud of gene histories, where within our species trees we have multiple gene histories, and then each one of these gene trees is present in, or can be present at different frequencies. In a perfect multi-species coalescence scenario, the most frequent one, or the most frequent quartet of 3 species plus and outward would be the true species tree. These papers, what they did is like they designed some methods of genomic interrogation, basically creating or testing or carrying out, hypothesis testing for each individual gene tree and seeing overall how many or which one is the most commonly accepted phylogeny across all the gene trees that we have. And in this case, it was this. Most of the gene trees in their paper actually recover monophyletic Cruciformes, which actually makes quite more sense than what we were getting. But this is all just to tell you that this is like how I came to realize like the difficulties of these of phylogenomic analysis, and how we do have a lot of heterogeneity in phylogenomic data sets. This heterogeneity could be due to biological processes such as incomplete lineage sorting or horizontal gene transfer, but also to non-biological reasons, like, for example, systematic errors. Just, for example, using non-accurate models to infer our phylogenies. And most importantly is that increasing or adding just more data may, in fact, reduce this random error, but could also result in increasing problems of systematic error and eventually obtaining very high support for wrong species trees. So in the end, we have a big challenge when we are working in or trying to use genomic data to elucidate phylogenetic relationships or evolutionary histories. The first one would be, what is the set of markers that we need to use? Is this set of markers useful? Can we recover these homologous markers across very many distant taxa? And also, are the models that we're using to in our phylogenetic analysis, in our inferences, are they accurate? And is there a way to get rid of genes that deviate from these models, or regions of these alignments that we can get rid of to adjust these models better? So the first example that I started working on, and because I was interested in this group before, when I was more interested in just phylogeographic and biogeographic patterns of fish, was the group of the Gymnotiformes. These are the Neotropical electric fish. This is a really cool group of fish that is distributed in South America and in Central America, and they have this outstanding capacity of detecting and producing electric fields. And there are 5 families in this order. Some of them produce electric pulses, and some of them produce electric waves. This is important because these families, and it's still not understood how these different wave types or pulse types have evolved and how they are related to each other. Even Darwin was fascinated by these electric fish. He mentioned this he briefly mentioned electric fish in his book of The Origin of Species, and was talking about the origin of these electric organs. That is like, oh, if the electric organs had been inherited from some one ancient progenitor, we might have expected that all electric fish would have been specially related to each other. Of course, he already knew that not all electric fish are related to each other. There have been many instances of electrogenesis evolving across the fish tree of life. But so far, within this radiation, the particular radiation of electric fish in the Gymnotiformes, there are many hypotheses that have been proposed. And not all of them recover like all the families that produce pulse-type waves as monophyletic or wave type as monophyletic. It depends on whether we're considering only molecules or molecules and morphology and so forth. So yet again, here we had an interest on resolving these interfamily relationship of Gymnotiformes within trying to figure out what is the evolution of this electrocommunication system, but also seeing if, again, genomic markers like ultra-conserved elements could solve this issue. In this case, what we did is we analyzed a data set of about almost 1,000 UC loci, and we carried our different phylogenetic inferences using, for example, concatenated data. And in this case, we recover a phylogeny that had already been proposed that recovered the pulse types, families as monophyletic, but not the families that produce waves. On the other hand, when we considered other inference methods like this, like in Astro, that we are incorporating or we're considering this heterogeneity due to possible incompleteness sorting, we do recover, again, high support for the monophyly of the families that produce pulses, but very little support or no support at all for these families that are producing signals in the form of waves. Again, where is the problem? The problem relies here at the crown node of all these radiation, this old radiation, but we have very, very short nodes. Again, all these problems that we had observed before could also be playing a role here, right? But also we have this problem of being an ancient radiation, so homoplasy, could be playing a role. Again, incompleteness sorting, or just, hey, our data is not informative enough. So far, many studies had been using different approaches to try to filter or select specific data or subsets of the data that fit better either the models or were more promising in terms of how informative they could be. And what had been done so far was, for example, okay, let's just pick among all these genes that we have sequenced, the ones that have more parsimony informative sites, or the ones that have a better fit to a clock-like model, or something like that. And this is what I call non-targeted filtering, right? So it's like, oh, let's just select markers that could be more informative for a question. But I thought that we could also try a different type of filtering that we call targeted filtering, that was based on the phylogenetic informativeness to solve a specific question or a specific relationship in our tree. And in this case, the relationship that we want to solve is here at the origin of the Gymnotiformes. And the way that we thought that we could do this is by plotting across the time scale of our ultrametric tree the phylogenetic informativeness of each one of our loci. Here, each one of these lines represents one locus, one of these ultra-conserved elements, and this is just the profile of its phylogenetic informativeness, basically the likelihood of a particular substitution to occur along time of the tree, and then we could integrate that across the whole phylogeny. So we can see that most loci are more informative in this area, but actually we are interested on loci that are informative in this area over here. So basically, very simply, what it is like, okay, let's just pick, for example, 50% of the genes that, or the loci, sorry, that could be or have more information at this time scale. And we did that, and we reanalyzed, and now we have half of the number of genes, and we reanalyze the data the same way that we did before, and the concatenated analysis resulted in the same phylogenetic hypothesis. Again, the wave type families are not monophyletic, but when we do an analysis of species tree analysis, incorporating or taking into account incomplete lineage sorting, we see that this value that was only about 50 before now is 88. Still not super high, but that is a big difference. And this is a novel hypothesis that has not been proposed before. Now maybe the wave type families could have a common origin. More interestingly than this is that when we were using like fully coalescent models or methods, sorry, like in bees, when we are actually at the same time inferring the gene trees and the species tree, the performance of this method when we were using the targeted filtering was way better than we were not using when we're not filtering the data, not only because we recover less consensus trees, but also because the most common one was in 98% of the gene trees recovered this most common topology of reciprocally monophyletic post-type families and wave-type families. So in this case, for this particular study, not only we show that a novel hypothesis for the evolution of this group of fish, of how these post-type and wave-type families could have evolved, but also show that maybe targeted filtering could be used to reduce some of the disagreement that we find among our gene trees and have better support or for our species trees. However, this method is not valid for all inference strategies. As we say, like concatenated analysis are insensitive to this type of filtering. Again, going back to the challenges that we might be faced when looking at a phylogenetic inference, we have seen how a little bit about how we could filter unreliable genes, but we could also look at what type of markers are better or could be more promising for solving different relationships. And everybody knows that different data sets may yield different results, either because we have different taxon sampling, because we have different data types, intrinsic biological characteristics of the data, whether they are coding, non-coding, faster evolving, etc. In phylogenomics, most of the time, what we're looking at is a reduced representation of the genome, even though when we're sequencing whole genomes, we're only picking or selecting parts of that genome to analyze those, not in all cases, but in many of those. And many of you would be familiar with this type of markers, whether they are some type of flavor of RAT-seq data, transcriptomes, or different methods of target capture, whether we are capturing exons or ultra-conserved elements, like in the case that we're looking at. But usually there are not many reasons to pick one of these type of markers. We rarely have the time or the resources to think, oh, should I use exons or should I use ultra-conserved elements or should I use this other thing? Maybe we're driven by just which one is cheaper, what is available in my lab, what other person in my lab knows how to analyze or to work with, or sometimes we do have really truly biological reasons that we might base our decisions on. But this is rarely tested, right? So here we posed these questions for an example of cichlid fish in the Neotropics. We all know how cichlids are just classic textbook examples of these of speciation studies because of their phenotypic behavior on just species diversity that they show. Most of them are based, or most of these studies are based on African groups, but also in the Neotropics there are interesting groups. Particularly here we were interested in the cichlids of Middle America, where most of them belong to this tribe of heroine cichlids. So we're interested in resolving these relationships among this group of heroine cichlids. And again, we were using ultra-conserved elements to try to infer these evolutionary relationships. And well, we did our thing, right? Our trees, we recovered some high support for all the groups in this tribe, but the phylogeny that we were recovering was contrastedly different from a recent paper that was using a similar approach, but using exons instead of ultra-conserved elements. And this was not, we would expect that different data sets are going to may result in different phylogenetic hypotheses, but this was relevant because this affected how we could reconstruct the biogeographical history of these groups, right? Basically, if we were using our phylogeny, we could infer, or one of the hypotheses of colonization would be a South American origin of the heroine, and then independent colonizations of the Nandopsis groups here in the Antilles and the rest of the heroines in Middle America. If we were using we were basing our inferences in the exon phylogeny, one of the possible hypotheses could be just sequential colonization through the Antilles, also could be through Middle America. Some people also propose a back colonization of South America. In the end, there are different inferences and different possibilities, different consequences of these phylogenies to understand the evolutionary history of these groups. And in their paper, Ilves and collaborators, the exon paper, they attributed these differences mostly to the inference methods, whether they were, again, either using multi-species coalescent methods or concatenated methods. So we said, okay, let's compare these and let's compare the efficiency and see if these differences occur the same when we are using also a different type of markers. And we thought this was interesting because by evaluating different data sets, this could help us identify these conflicting regions. So for example, regions that are recovered with high support with different data sets, but also because understanding why these differences occur may help us just identify what type of marker might be better for a particular situation. So what we did is like, okay, we had a similar data set in terms of species for our UCE paper and the Exxon paper. Ilves et al. Luckily had shared and made publicly available all the data, so we could just make it fit and make it comparable with our analysis. And what we found is that not only there are differences due to the inference method, but also there are differences due to their genomic markers. So basically, what this means is like UCEs, for example, always, regardless of the method, always recover some relationships that were not recovered in the exons, and vice versa. When we're looking at inference methods, that regardless of UCEs or exons, maximum likelihood recovered some relationships that were not recovered with multi-species coalescent. And also, there are temporal biases, right? So there are markers that recover high support for shallower relationships, whether others are recover higher support for deeper relationships. So we investigate a little bit more about the characteristics of this information in this type of markers. And what we observe in general is that exons in general are much more informative than UCEs across the whole tree of life. But of course, there is a lot of variation, right? So this is these profiles. This is the average of all these phylogenetic informative profiles that I had been showing you before. But you can see that this means that there are markers that are as low as this, and other ones are very, very informative, and UCEs are lower across most of the time scale of our tree. This would be where this origin of the tribe and the subfamily would be located in this time scale of our gene tree. Sorry, our species tree. And looking at the gene trees, what we see are also quite a few of these differences. On average, again, the concordance of the genes is much higher for the exon gene trees than for the UCEs. So in general, these exons have more trees or more gene trees that agree with the final species tree than with the UCEs. But this is not what happens when we're only considering the sites, the substitutions across the sequence. This could be due to UCEs being shorter. When we're looking at them individually, they perform worse. But when we're looking at the whole sequence, there is basically the same amount of information or useful information in both cases. Based on this distribution of the frequency of these gene trees, we could also see how UCEs are in general, more affected by processes like incompleteness sorting, but the exons show a lot more differences in the frequencies for alternative topologies. But this is only for this set of relationships in here. It varies a lot across the whole tree. Here is also a different way of showing you this information that we have for these same relationships across UCEs and exons. Each one of these little circles is one of the loci that we are looking at. And here what we're plotting is the gene-wide log likelihood score. Basically what we're looking at is how different in support is each gene, how different the support of each gene towards the species tree topology or an alternative topology is. So if the value is positive, we see that this locus is supporting the species tree. If it's negative, we say that an alternative topology is better supported. And we can see here what we obtain for UCEs and for exons. And despite that we can see here a lot of dots here in positive values, actually most of these genes and most of these bubbles are around this zero line, which basically is telling us that these genes are telling us nothing. They are not significantly better supporting individually, either supporting the species tree or the alternative topology. So there's very little information in general in all of them. When we look at them at the average of all these markers for different relationships, here, what I want you to look at, this is again the same gene-wide log likelihood score, is the difference in scale between this exon data set and the UCE data set. And what we see is that exons, in general, have more outlier loci. So they have more loci that either support very much the species topology or an alternative topology. So there are genes that are very extreme in the amount of information, whereas UCEs are more noise and, in general, very little information in all of them, but very few outliers. Oh, one important thing here is that if I removed these outlier loci from the exons, now the remaining exons, what they give you is the topology of the recovered by the UCEs. And so there might be just like a disproportionate effect of just a handful of loci in the exons that does not exist in the ultra-conserved elements. But in the end, just two very obvious conclusions is that relationships that are unstable are just hard to solve regardless of the data. That's not surprising. And also, in this case, this set of markers just provide just as good as an estimate for a phylogeny as the other. So in the end, there's not much. They don't say much, either of them. Sad conclusion, but this is what we're getting. All right, so we have looked at filtering out unreliable genes, evaluating different types of set of markers, and let's look now about how different models or methods to evaluate relationships can affect our results. And yet again, we're going to look at another example of cyclids. And as I was saying earlier, most of the work that has been done in this group comes from examples in the East African lakes, where the major drivers of this diversification are usually adaptations and hybridizations as well. On the other hand, in the river cichlids, vicariance is usually invoked as one of the main drivers of diversification. But recent papers, for example, here with Naoko Kurata, working in fish from the Congo River, have also proposed how in some cases, ecological or other climatic drivers, or even adaptations, could also be major drivers of diversification in rivers. And Naoko has worked mostly and her group, mostly on the Congo River. And the Congo River is particularly interesting both for its, its size, first and foremost. It's the second longest river in the world. It crosses the equator twice, so there's a lot of rainfall, and there's always a high volume of water in this river. The area that it drains is huge. It basically encompasses most of the Democratic Republic of the Congo. Here we have the East African lakes here, Lake Tanganyika, that is connected to the Congo River. But one very interesting part of the Congo River is this small stretch at the end of the river. These 350 kilometers that it only is about 2% of the area of the whole drainage has a disproportionate amount of the diversity of fish in this river. Of course, the Congo has the amount of fish or fish species, and particularly cichlids, compared to Lake Tanganyika is very small, but 30% of the cichlid species in the Congo River are in this lower part, here from this lake-like feature that is called Pool Malebo, until its end on the Atlantic Ocean. But also, not only the number of species, but also how many, or the highest percentage of endemic species are also in this region. And what is going on here that makes it so species rich? Well, it's all about the geomorphology of this river. Above or upstream Pool Malebo, this is what's called the Upper Congo River. The surface is very flat. The river is just like flows slowly, meanders, sandy bottoms, and just very homogeneous in terms of habitats and the features of the lake. But in the lower Congo, from Pool Malebo to the Atlantic, the drop of this river is very dramatic. It just like drops almost 280 meters in this short distance. It forms crazy rapids, gorges, very, very deep regions. And particularly, there is a short stretch of about 15 kilometers that are called the Inga Rapids, where the river drops over 150 meters. So it's hard to picture in just a photograph how huge and how dramatic or how powerful this hydrology is in this lower part of the river. Actually, there are two of the largest hydropower plants in the world are here, and there is a project for third one and so forth. But that's another story. So not only these powerful hydraulics have played a role in separating and creating barriers within the lake, but also in this region, this power of this hydrological power has carved the river, making it the deepest river in the world. There are regions that are above 200 meters deep, and there's a lot of there are several examples of just these cave-like phenotypes of different families that occur in this lower part of the river. Some species of in families of catfishes, also these spiny eels, mormyrids, which is another radiation of electric fish, the African radiation of electric fish, and also cichlids. In particular, the only known blind cichlid that we have, that is Lamprologus lithops. And what is interesting is that Lamprologus lithops is only known from dead or moribund specimens that just like surface once in a while at particular spots in the river. So it's kind of unknown what is its real distributions, how many they are, where they live, but we assume, or people that know a lot more, assume that lives in these deep gorges in the river. However, because we don't know its distribution, we don't know what is its relationship with the other species of Lamprologus that also live in this region. Most of the species of Lamprologus, and in general the tribe Lamprologini, occur in Lake Tanganyika. There are over 200, there are hundreds of species of this group here in Lake Tanganyika, but there are only 10 species out of the lake in rivers, and 9 of them belong to the genus Lamprologus. Some of them are widely distributed, most of them, the ones that occur in the upper Congo River, in this lazy river flowing, and other species have much more restricted distributions, and those are the ones that occur here in the lower parts of the river. Here, as we can see, this is the profile of the lower Congo. Here, this is the region that I was telling you where this short stretch of the Inga Rapids where the river dramatically drops. But we can see how a lot of these species have parapatric distributions along the lake. And in this region is where lithops has been found, right? Just like what it has just popped up randomly in the lake. We don't know what is the relationship of these species, even what is the relationship with the lake species. It's unknown, or there are different hypotheses whether the river has been seeded from the lake or the Tanganyika radiation was seeded from the river. Here in this just small phylogeny, but including all the species, where a student was doing his TFG, I guess, on using mitochondrial genomes of these species. What we find is that not even species are not even monophyletic when we're looking at their mitochondrial genome. And here in red are species from the lower Congo, and in blue are species from the upper Congo. There's also not a good separation between them. So they're all mixed together. It's a mess. There's little that we could understand from this. However, also in a recent paper that included most of the Tanganyika radiation, looking at the lower of the Congo River Lamprologus, what we find is like, oh, lower Congo River cichlids are reciprocally monophyletic from the upper Congo, but again, very low support, or at least a lot of uncertainty in these relationships. Here, the red pies indicate that the gene concordance is very low, and also the red branches indicate that what we obtain using concatenated or multi-species coagulation methods are very different. Well, what they obtain. So there's a lot of uncertainty whether the lower Congo River is actually a monophyletic group. So here, what we try to do is, like, given that so far most of the phylogenetic methods had been unable to provide a solid hypothesis for these species, we try to, okay, let's look at a more broad scale, or let's take a more broad scale approach and look at not only phylogenomic, but also population genomics or phylogeographical approaches to try to discern these relationships. And most importantly, see if we could differentiate whether these alleles that are being shared between species is just due to ILS, or actually they are hybridizing, because this is one of the possibilities that Astudillo Clavijo had proposed in their paper. What we obtain is that, again, yes, we do find the Congo River nested in Lake Tanganyika, so it seems that the river was colonized from the lake, and we also obtained reciprocally monophyletic species from the lower Congo and the upper Congo, but again, with low support. The species tree basically showed very similar relationships. Again, all these red circles are nodes that are conflictive because we cannot basically reject that these branches have zero branch length, which means that it could actually, based on our data, they could be resolved or be replaced by a polytomy. What is important about this data set and about the data that we're using to construct the species tree is that the gene trees that we're using have almost no information. The average bootstrap of these gene trees is 11. So basically, what is this? What are these loci telling us? How can they recover this if individually they have such little information? Well, what we observe is that there is a lot of variation here in these quartet scores. Basically, it's the frequency of the quartets that we're observing in the gene trees compared to the species trees. Some of them have up to 75% of the quartets are also present in the species tree, but for most of them, they're around this 33%, which is basically any possibility could happen. What is interesting from this graph is that the differences in how much support these gene trees have for the species tree topology does not depend on the amount of gene trees that we have. Here we can see that for all the nodes, the number of gene trees that we could use or that were useful or informative was basically the same. So there should be other reasons for these differences in signal. The signal was there because when we're using some different population genomic methods, we could see that the lower Congo was separated from the upper Congo, at least along this axis, but this axis also was showing that how there are species like lithops, again, the blind cichlid was highly differentiated, also other populations that were highly isolated. And again, this is kind of like similar to a structure plot, but an admixture analysis also showing and supporting the difference between the lower Congo and the upper Congo. Taking advantage of these genomic data and how we could create linkage groups and see how these linkage groups have been inherited between different species and to see how much of this shared co-ancestry they have. Again, we kind of have support for these reciprocally monophyletic upper Congo and lower Congo, how most of the species within the lower Congo are more related to each other than to the upper Congo and vice versa. But one important thing are these two species, Lamprologus werneri and Lamprologus congoensis, werneri being lower Congo species that is more related to some of the upper Congo, and congoensis the other way around, being more related to these lower Congo species. So what is going on in here? Going back to this mitochondrial phylogeny that we were seeing before, we could not find a clear answer because we were not able to extract the whole mitochondrial genomes from our raw reads from the genome, but we were able to get at least one mitochondrial genes for all the species. And what we find is that the lower Congo and the upper Congo form two clades that are non-sister to each other but are nested within Tanganyika. So I call these mainly lower Congo and mainly central upper Congo because there's a lot of mixture, right? So there's a lot of species here from the lower Congo that are also present here and vice versa. But there are two important things. Werneri and congoensis, which were the species that I was highlighting before, This is the only lower Congo species that is only present in the upper Congo clade, and Congoensis, vice versa, is the only upper Congo that is present in the lower Congo. So what is this? Is this like two reverse cases of mitochondrial capture, or what is going on in here? This looks like a little bit messy, but it makes more sense when we look at it in the map. Here with squares, I have species that, or have indicated species that belong to this mitochondrial clade of the upper Congo, and with circles, I have species that have mitochondrial DNA from the lower Congo. Regardless, remember that all these form a monophyletic group when looking at the nuclear genome. So there is a phylogeographic structure for the mitochondrial DNA, and this is still an open question of what has happened in here. Is this colonization from a hybrid swarm from Lake Tanganyika? Are these all ancestral polymorphisms? But how, if that was the case, how was this phylogeographic structure maintained? Did they colonize independently, and then there was gene flow after colonization? I'm more inclined for this second option because our hybridization analysis show quite a lot of instances or possible instances of hybridization. Here I'm only showing you the most, or the ones that show the highest fractions of the genome that could be of hybrid origin. But here what is more interesting is that most of these hybridization events do not occur within the lower Congo or within the upper Congo, but between these two branches, right? Between the branch that includes Werneri and the branch that includes Congoensis. Even though, in most cases, predating the speciation within those. So it could be that there was hybridization, and then this hybrid genomes then speciated due to the development of the Congo River and these rapids and all these crazy ecological conditions and geographical conditions that occur in here. So in the end, what we can see here is that maybe looking at only one specific phylogenomic approach might be hard if we are dealing with low informative genes, because most probably because of the difficulty in modeling these evolutionary models for these markers, and a combination of inference methods could help us discern what are the reasons of incongruence, whether it's ILS, whether it's possible hybridization. Why are all these things relevant now that we're for other cases beyond FISH or beyond UCEs and beyond exons? In the end, when we're looking at sequencing whole genomes, I think we should ask what type of data we should extract from these genomes. Is all the data equally useful, and how we should combine it, and how we should analyze these different data sets. And in the end, full circle, I feel like we are ending up in this same previous picture when, again, phylogenomics is promising, resolving a lot of the difficult nodes, but there's still a lot of challenges and questions that we need to address, and we should focus more on looking at this information in the data rather than just testing the hypothesis and trying to fit them to the sequences. All right, so I would like to thank many of the co-authors and collaborators in all these projects, the museums that have provided tissues and data, and people have provided photographs for this presentation, and thank you for listening.
Thank you, Fernando. So we have now a bit of time for questions. Está un poco alto esto, ¿no?
Hi, thanks a lot for the talk. It was great. I have a question about, so one thing that affects phylogenetic signals a lot is recombination rate. Mm-hmm. Do you have a sense of whether UCs or exons are like more in low recombination rate regions, or the contrary, or no trend at all?
Yeah. I don't have data from my own studies, but there's data from other comparative analysis of how UCs are distributed along the genome. In general, they do not show more recombination than other types of markers. They are usually distributed quite randomly across the whole genome. What is more interesting about the and I don't know if this is related to what you're asking, is usually they're affected less than other types of markers by duplications and other type of paralogous copies. There's a problem of paralogy, particularly in fish, given genome duplications and things like that. But in terms of recombination and that leading to also possible duplications and so forth, they're not as problematic as other types of markers. But yeah, it's not one of the main problems of UCs, recombination.
Okay, thanks.
Do you have any other question? Se lo doy a Fernando, que es mayor.
Thank you, Fernando. This was amazing. But please tell me that you also work in something easier, because this looks extremely challenging. And I wonder if you've thought about using synteny now that we're in this whole genome sequencing or chromosome-level sequence era. Do you think synteny or conservation of synteny could help you understand what is happening here?
Yeah, probably. Yeah, that's a great question. And actually, one of the things that we see when we're comparing this is rather than increasing the number of loci, usually probably increasing the length of the sequences that we are that we're analyzing, and having chromosome level sequences or a scaffold, or being able to put that in the genomic context could help us more, rather than collecting a lot of data from many different chunks, because what we see is that is not the problem of the amount of data, is just how these noisy independent signals. So yes, that would be a great question, and I guess that's also like a technology budget issue rather than being able to do it. But yes, probably that would help a lot, yes.
Thanks, Fernando. It was a great talk. I really enjoyed it. I have a question, two questions. One related to what Lois was asking about the paralogy of UCs. Is there a way of teasing up, putting apart paralogy in UCs? I have no clue how this works.
I think all the methods, and I'm not very familiar with those methods, but I think that all the bioinformatic methods to detect paralogy are not very reliable, and there's nothing specific for UCs. But I'm not an expert on those.
Or maybe they are not affected by so much affected by paralogy. I don't know.
They are, and we have had that issue come up with. We have not tested it, and for example, Brand Fairclough, he says that is not an issue, but I know that he has dealt with that with reviewers asking him to address those things, and in the design of the probes, paralogy is taken into account, but other than that, I don't know whether they are more or less, or that's a big issue. To be honest, I don't know.
Okay, thank you. Second question, I really like a lot this paper in GB where you compare different methods to resolve the same phylogeny. How generalizable do you think is this kind of, or how? So can we use the conclusions of one particular group on comparing different methods to other groups, or is it very question specific?
Yeah.
I mean, sorry, because you mentioned that there are very few studies that compare the different methods for the same group. Why? Because you have to sequence the things twice.
Yeah.
So it makes sense that there are not many studies. But I also wonder if we can actually, so if we had like 100 studies, then probably you can see whether the pattern is common or is different. But
yeah.
What is your feeling about this?
I think it's both. I think both. I think there are some things that could be generalized, but there are other things that are very question specific, particularly because of the time scales that we are interested in. Sometimes we are, as I don't know, the scale that you work is way more ancient than when we're looking at, I don't know, a radiation of African cichlids or things like that. So the scale is is one thing, and that limits how we can generalize these conclusions. But there are other things that are common across things. Recently, I was reviewing a paper that was also doing this type of comparisons, and kind of came to the same conclusion in terms of how how informative some type of markers are compared to the others. In particular, how exons have a tendency to over-support either the species tree or alternate topologies, whereas other methods are less informative but more common in their lack of support. So I think that could be generalized. How that affects each particular scenario, I think that is that might be different. Yes.
Cool. Thank you.
Thanks for the talk, Fernando. I was, for the first paper or the first study that you show us, the Gymnotiformes, I was wondering the morphological implications of the two alternative hypotheses that you are, at the end the notes are short, so you can play around with the data, but then it's difficult to describe one or the other. And when you have the paraphily of the wave type, basically, then the ancestor of all Gymnotiformes might have a wave-type organ.
Yes.
But when you have the two monophyletic, reciprocally monophyletic, then the ancestor might have one or the other, or none of the two, and something else.
Yes.
So I guess morphology is not that easy to create or to generate, to evolve an electric organ. So I'm wondering how much the interpretation of your phylogeny is supported by the morphology.
Yeah, that's a great question. And there's also not a complete agreement with what is the ancestral state of the electric organ. In the end, the electric organ is a modified muscle cell that just is able to produce all these electrocytes to trigger all together and produce this electric signal. In theory, even though there's also different interpretation of this, the pulse type should be a more the ancestral state than the wave type. Just because of but this is because of the embryological origin of the electric signal. Wave type families embryologically, I think, start, or the embryological electrocyte is more similar to the pulse type electrocyte, and then develops and differentiates in the wave type. So for some, that supports that the pulse type should be or could be the ancestor. Also, what is most commonly accepted is that the catfishes are the sister group of the Gymnotiformes. And even though catfishes do not produce these electric signals, they do have similar embryological structures that are more alike the pulse, but is not clear. Yeah. And also there are some morphological hypotheses that say that the wave type is is the ancestral.
So
Yes.
we are agreeing with the paraphyly.
Yes. Yes. Yeah.
Oh, just one last question, and then we go for beer. So when you compare the ultra-conserved elements with the exons, what I see there is a lot more information in the exons, perhaps not for reconstructing this phylogeny, but perhaps useful for inferring other things. Like, this might be a very long way to get into candidate genes for some interesting speciation events or evolutionary events.
Yes, exactly. And actually, for example, that paper that we were comparing it to was part of a series of papers of this group that were also looking at opsin genes and other candidate genes that might be directly involved in speciation or in reproductive isolation of some of these groups. Yes, so there are different implications of how useful these or what other additional information we could get from these markers. Again, also how the selective pressures that these markers could have. Not that UCs are not under selection, because probably they are, given that they are ultra-conserved in many taxa. But yes, yeah. So there is an additional utility for exons that for UCs, because their function is not so well known. Yeah, it would be lacking there, yes.
Okay, so unless there's a very quick question, we're going to stop it here. So thank you again, Fernando, for this very nice talk.